Alright folks, so once glycolysis is finished, right, in the presence of oxygen, your pyruvate is going to end up moving into the mitochondria. And remember, this enters then the second stage of cellular respiration. However, the second stage, remember, is sort of divided up into two parts. Before the pyruvate can go into the citric acid cycle, which is the bulk of the second stage of cellular respiration, it needs to be modified a little bit. And here's where we go into sort of an entry step first before going into the citric acid cycle. This entry step, remember, is called pyruvate oxidation. Alright? So when the pyruvate moves into the mitochondria, it goes through a three-step process that is catalyzed by a large multi-enzyme complex that will take it through the loss of one of its carbons. Right? So the pyruvate, we started out with 3 carbons. After the loss of one of those carbons, as carbon dioxide, we're left with just 2 carbons. Those 2 carbons have a few of those high energy electrons taken away, and those electrons are given then to NAD+ to form NADH. And on top of that, we also have a hat added onto those 2 left over carbons from the pyruvate. Once we add that hat, we've formed our molecule of acetyl-CoA, and then the acetyl-CoA is ready to go into the citric acid cycle. All right, so when we go into the citric acid cycle, this is going to complete breakdown of the whatever is left of glucose, right? Those last 2 carbons. If you've taken biology before, you may have heard of the citric acid cycle being referred to as the Krebs cycle. One and the same, so you can call it either way. For the sake of our class, we're going to be calling it the citric acid cycle. So as the acetyl-CoA enters the citric acid cycle, remember it's a cycle, so whatever molecules you start out with, you have to essentially regenerate at the end of that cycle. For each glucose molecule that goes in to cellular respiration, remember glucose is 6 carbons, right? And in glycolysis, we end up splitting those 6 carbons into 2 3-carbon pyruvates, and each of those pyruvates will then be changed into acetyl-CoA, so both of them will lose one of those carbons, leaving 2 carbon molecules left over. And then each one of those is going to enter the citric acid cycle. When the first one goes in, we'll make 1 ATP, 3 NADHs, and then 1 FADH2. And the second one, when it goes in, it will do the same thing. So essentially, even though every turn of the citric acid cycle produces 1 ATP, 3 NADHs, and 1 NADH2, for one glucose molecule, we end up doubling that output because we have the citric acid cycle taking 2 loops around, one for each of the acetyl-CoAs. Okay, so let's take a look at the citric acid cycle. Now, this slide summarizes essentially the citric acid cycle, and this is what I want you guys to focus on. On top of that, I'm also going to ask you to add a little molecule right at the beginning step, so kind of where 11 o'clock is on the clock, right? There's a molecule there called oxaloacetate, and this molecule is a 4-carbon molecule that's going to be linked to the 2 carbons that come in from the acetyl-CoA. So as the acetyl-CoA goes in, you'll notice that the coenzyme A hat gets removed, and then once that gets removed, the 2 carbons from the acetyl-CoA get linked to the 4 carbons from the oxaloacetate, and the rest of the cycle is really the degradation of the remaining carbon molecules, extracting as much energy as possible. We make some ATP, we take away some of those high energy electrons, and then we end up regenerating that same oxaloacetate 4-carbon molecule so that we can start the same cycle all over again with another acetyl-CoA molecule. So as you're studying, focus on this slide to get the general gist of the citric acid cycle, plus add in that oxaloacetate, because that is a very important molecule, because that's what we start out with to link to the acetyl portion of the acetyl-CoA, and that's what we have to regenerate at the end of the citric acid cycle. Okay, so the citric acid cycle essentially has those 8 steps then. And remember, just like every other chemical reaction within a cell, each step is likely going to be catalyzed by a specific enzyme, which is definitely the case for the citric acid cycle. The very first step is going to be that most important step, and this is where you have the 2 carbons from the acetyl-CoA joining with the 4 carbons of the oxaloacetate. And then the rest of the cycle, steps 2 through 8, then are going to be essentially getting closer and closer into regenerating that same 4-carbon oxaloacetate. At the end of the citric acid cycle, the majority of the energy that was left over and what was left of the glucose as it came into the citric acid cycle now gets removed from that acetyl molecule and stored as those high energy electrons in FADH2 and NADH. Remember, those electrons are then going to be shuttled to the third stage of cellular respiration, where we have the energy of the electrons and cashed in essentially to make ATP. Alright, so let's go through then the individual steps of the citric acid cycle. And here's the very first step, that probably most important step, where we have the acetyl-CoA molecule coming in with its 2 carbons. You'll notice that those 2 carbons are shown in red, just to highlight, hey, these are the carbons that are coming in from the acetyl molecule. And the 2 carbons from the acetyl coenzyme A, once they lose the coenzyme A hat, those are going to be linked with the 4 carbons from oxaloacetate, right? And this is going to make essentially another 6-carbon molecule, and this molecule is called citrate. Citrate then is going to be reorganized a little bit into its isomer, isocitrate. And the subsequent 2 steps is where we start to now degrade the molecule. Think of, if you're a thief, you're not a thief, but if you were a thief, and you're going to strip a car, right? You're going to take away all of the expensive parts of the car to use to sell for parts, etc., so that, you know, you get some money. We're going to do the same thing to this molecule of isocitrate. We're going to start just stripping it, so we end up with bare bones carbon dioxide, and we've extracted as much of its energy as we possibly can. So the next step, isocitrate, is going to lose some of those high energy electrons. Those are going to be given to NAD+ to form NADH. We're also going to lose one of the carbons as carbon dioxide. If you take a look at that slide, that carbon, remember, is blue. And so if you look back, sort of trace back where that blue carbon came from, it actually came from the oxaloacetate. So this carbon didn't come in as a carbon as the acetyl group that came in during this cycle, but it's an old carbon that had come in at some point previously in one of the previous acetyl CoA molecules. An interesting thing to think about. So now we've gone from a, 6 carbon molecule, we've lost one of the carbons, we're down to 5. The next step is going to be essentially a repeat. We're going to take some more of those high energy electrons, and we're going to lose now from 5 1 more of those carbons. We're getting closer to our 4 carbon oxaloacetate. The remainder of the steps is really going to be remodeling that 4 carbon molecule. We're going to make some ATP. Remember that the making of ATP, again in the citric acid cycle, is going to use substrate level phosphorylation. So we're going to take the phosphate group from a substrate molecule and then donate it to an ADP to make ATP. Okay? And then the remainder of the steps, right, we're already at 4 carbons. We're just going to rearrange the molecule. We're going to donate some of those electrons to FADH2. We're going to donate some of them to NADH. And at the very end, we're going to end up regenerating that 4 carbon oxaloacetate molecule so that we can take yet another swing around the block. One more loop around the citric acid cycle with another acetyl-CoA molecule coming in. Okay, once the citric acid cycle is complete, remember we've made a little bit of ATP, right, shown at the bottom of the slide, and glucose now is completely degraded to carbon dioxide. All 6 of those carbons that originally came in as glucose, they have, sort of one at a time, been lost as carbon dioxide. So it's gone at the end of the citric acid cycle. But we've only made, a little bit of ATP, 2 from glycolysis and 2 from the citric acid cycle. That leaves 28-ish more ATPs for us to produce. Where is all that leftover energy now that glucose has totally been degraded? Well, remember, NADH and FADH2 are still holding on to those high energy electrons for us. We need to take those now and funnel them into oxidative phosphorylation, that very last step of cellular respiration, and cash in the energy that those electrons have to make ATP. Okay, so a little bit of a review here for this slide. Remember that when we finish glycolysis and the citric acid cycle, it's really the NADH and the FADH2 that are carrying those high energy electrons that accounts for most of the energy that has been extracted from that glucose molecule or from your food. Those 2 electron carriers, what are they going to do with those electrons? Well, they're going to take them, remember, to the electron transport chain, right? That series of proteins. So it is going to take those electrons, play hot potato with those, allow them to give up some of their energy, and then the energy that they give up ends up powering ATP, and this process is called chemiosmosis, and we'll talk about it in one of the next slides. In total, taking the electron transport chain plus chemiosmosis, this is actually the whole process of oxidative phosphorylation, that very last step in cellular respiration. Okay, so let's take a look now at oxidative phosphorylation. This particular image is going to show you the proteins that are involved in the entire process of oxidative phosphorylation. It's actually divided into two sections, and as you'll note on the slide, number 1, labeled down below there, this portion is going to be the electron transport chain. And then off to the right there, labeled number 2, that's going to be your chemiosmosis. So both of these processes, when put together, they are going to be now your oxidative phosphorylation. Where are all these proteins located? Well, you'll see a membrane there, right? This membrane is actually part of the mitochondria. So remember, think back to chapter 6 when we were talking about various organelles that are found in eukaryotic cells. Well, the mitochondria is one of those organelles, and it has that nice, smooth outer membrane, and then it has that highly convoluted inner membrane. Remember, we had mentioned that inner membrane, it's a good thing that it has all those cristae, or all those folds, because within that membrane, embedded in that membrane, are all those proteins that play a role in making ATP for the cell. So the more of that membrane you have, the more ATP you can produce as a cell, the more energy you'll have to power whatever cellular work you need to power. Okay, so if we take a look then at this particular image, this membrane is going to be that inner mitochondrial membrane, the one that's really folded. Below it, in light orange, this is going to be the matrix, the very inside of the mitochondria. And then on the outside of the membrane, that's going to be that intermembrane space. So that space in between the outer membrane and the inner membrane. Okay? All right, so all of these proteins, both that participate in the electron transport chain, as well as in the process of chemiosmosis, they're all embedded or at least closely associated with that inner membrane of the mitochondria. Now, the first part of oxidative phosphorylation is when the electron carriers, NADH and FADH2, get to the electron transport chain and drop off those high energy electrons. Now remember, those high energy electrons, right, they're going to have to bounce down that electron transport chain and giving up some of their energy that's eventually going to be used to make ATP. So let's take a look at that electron transport chain. On the left-hand side of this figure, you're going to see large purple protein complexes, and they're labeled with Roman numerals. So there's protein complex 1, there's protein complex 2, 3, and then 4. And then you'll notice that in between there are some smaller protein components. So there's protein Q, and then there's also site C or cytochrome C. And these are your movable protein transporters. So they'll pick up electrons, let's say, if we're looking at Q. Q will pick up electrons from complex 1 or 2 and pass them on to 3. And cytochrome C, or site C, will pick up electrons from the protein complex 3 and pass them on to protein complex 4. So if you take a look at where NADH and FADH2 are in the lower left-hand corner, they're going to drop off their electrons at the first series of protein complexes, and then the electrons are just going to move all the way through, right, bouncing all the way down until they end up at the very bottom of that electron transport chain in the lap of oxygen. Remember, oxygen is going to be that final resting place for the electrons. As they've gone through that whole entire electron transport chain, they've given up all their energy, they're done, they're exhausted, they fall into the lap of oxygen. Oxygen will also pick up some of the protons from the solution, and you will have the formation of